Have you ever found yourself standing in the grocery aisle, maybe clutching a protein bar, and wondering what truly happens inside your body when you consume it?
Mm-hmm.
Or perhaps you've pondered why some diets seem to emphasize certain nutrients over others, even claiming some are essential while others aren't. It's a common thought, really.
It is. And it hints at a far deeper, more intricate world operating within us.
Today, we're going to peel back the layers of that world.
Indeed. What seems like a simple question about daily nutrition quickly leads us into the fascinating, complex realm of amino acids. These aren't just abstract building blocks, as they're often portrayed. They are dynamic, incredibly versatile molecules.
Right, not just passive bricks.
Exactly. They play surprising and absolutely critical roles in everything from maintaining the robust structure of our bones and skin to, well, powering the most vital enzymes that keep us alive and thriving. They're orchestrating the very chemistry of life.
And today, that's exactly where we're heading. Our mission for this deep dive is to truly understand the world of amino acids. We're embarking on a journey to distinguish the must-haves, the ones your body absolutely cannot make, that must come from your diet.
The truly essential ones, nutritionally speaking.
Yes, exactly. Distinguish those from the make-yourself-at-homes that your cells are constantly churning out through these, well, ingenious biochemical pathways. We'll uncover some surprising processes, like how certain amino acids are modified after proteins are built, or even how our genetic code can be elegantly reprogrammed, you could say, to insert rare vital components.
It's quite amazing stuff.
Our goal is for you to walk away from this deep dive with the true mastery of this information, thinking about it with the precision and depth of, say, an elite professional in the field.
And to achieve that level of understanding, you should know that this deep dive is rigorously grounded in detailed, authoritative biochemical research. We've synthesized a comprehensive look at this topic, drawing from the very foundations of human biochemistry to give you an authoritative and truly comprehensive perspective.
Okay, let's unpack this fundamental distinction then. When we talk about essential amino acids, it's not as straightforward as it sounds, is it?
Not at all.
There's a critical nuance here that can really trip people up, leading to a lot of confusion in general discussions about diet and health. You're essential, and you might immediately think, oh, I need this one, and maybe I don't need that one quite as much.
Right. Or maybe not at all.
But that's not quite right when it comes to the human body's internal chemistry, is it?
You've hit on the core misunderstanding right there. It's crucial to understand that for human subjects, all 20 common amino acids are metabolically essential to ensure health. Let me emphasize that.
Yeah.
Every single one is needed.
All 20.
No amino acid is truly non-essential in terms of its vital role in the intricate machinery of the body. Every single one has a job, a function it performs to keep our systems running optimally, from building muscle to forming critical neurotransmitters. The issue isn't whether they're needed.
It's how we get them.
Precisely. It's how we acquire them.
So that's where the precision comes in. It's not about their importance per se, but rather their source, whether our body can make them or if we must get them from our food. So what's the correct, nuanced way to classify them then, if all 20 are metabolically essential?
Well, the key distinction lies in the terms nutritionally essential versus nutritionally non-essential. When we say nutritionally essential, and that term has sort of replaced the older, simpler essential, just to be more precise,
Makes sense.
we're referring to the 9 amino acids that must be present in the human diet. Our bodies simply cannot synthesize them, or at least not in sufficient quantities to meet our physiological demands, especially during periods of growth.
Okay, the 9 must-haves from food. Which ones are they again?
They are histidine, isoleucine, leucine, lysine, methionine, phenylalanine, threonine, tryptophan, and valine. Now, you might sometimes hear arginine referred to as semi-essential.
Right. Why semi?
Because while adults can synthesize some of it, children often cannot produce enough to support their rapid growth and therefore require it from their diet. So it's conditionally essential, you could say.
Got it. So these 9 or 10, depending on age and context, are the ones you absolutely have to eat because your body doesn't have the cellular machinery to build them from scratch. It's like a factory that only produces certain parts.
Exactly. And for others, it relies entirely on external suppliers, your diet.
What about the others then? The nutritionally non-essential ones, how many are there?
The other 11 amino acids fall into the nutritionally non-essential category. These include alanine, asparagine, aspartate, cysteine, glutamate, glutamine, glycine, proline, serine, and tyrosine. Oh, and hydroxyproline and hydroxyleucine are often included here too, though their story is a bit different, as we'll see. Now, while these are also, and I'll reiterate, metabolically essential, meaning they are vital for various bodily functions, they need not be present in the diet.
Because we can make them ourselves.
Exactly. The human body possesses the complete enzymatic machinery and metabolic pathways to synthesize them from other precursors readily available within our own cells. It's a remarkable testament to our body's self-sufficiency, really.
That's a crucial clarification.
Yeah.
It makes so much more sense when you frame it as dietary requirement versus metabolic necessity. And this distinction wasn't just, pulled out of thin air, was it? How did scientists even figure this out back in the day? Especially that specific list.
Oh, no, definitely not thin air. It was a meticulous and frankly pioneering effort, primarily established back in the 1930s. Researchers, most notably William Rose and his team, conducted groundbreaking biochemical investigations.
Oh, what did they do?
They fed human subjects diets where purified amino acids completely replaced whole proteins. Imagine the dedication required for those studies.
Wow, yeah. Highly controlled.
Extremely. By carefully observing health outcomes, growth patterns, and metabolic responses of individuals on these highly controlled diets, they could determine precisely which amino acids were absolutely necessary to include in the diet to prevent deficiency symptoms and support normal health.
So they systematically removed one amino acid at a time, basically.
Essentially, yes, or varied the combinations. These pioneering studies paved the way for subsequent biochemical investigations that ultimately revealed the precise biosynthesis pathways for all 20 amino acids. It was a remarkable feat of scientific detective work, laying the foundation for modern nutritional science.
Okay, here's where it gets really interesting for me, and a little counterintuitive, perhaps.
Yeah.
Why would our bodies evolve to lose the ability to make something so vital? You'd think that if something is essential for survival, the body would want to keep the blueprints, right? Keep making it internally.
Right. Why delegate that responsibility to what you happen to eat? It feels like putting all your eggs in one basket, doesn't it?
Exactly.
It's a fantastic question, and it points to a really elegant principle of evolutionary efficiency and adaptation. The intriguing concept here is that dependence on an external source for a specific nutrient can actually be of greater survival value than retaining the ability to biosynthesize it.
Greater survival value. How?
If, and this is the key condition, if that nutrient is consistently available in ample quantities within the environment and the typical diet.
Ah, okay. So if it's always around anyway.
Then it becomes a strategic trade-off. Think of it this way. If a particular amino acid is readily and consistently available in the food sources our ancestors consumed, why should an organism expend precious energy, ATP, our cellular fuel, and other resources to maintain and express the complex genetic machinery required to synthesize it internally?
So it's like a biochemical cost-benefit analysis playing out over evolutionary time. If it's easy to get from the outside, ditch the internal factory and save that energy for something else.
Yeah.
That makes a lot of sense, actually.
Precisely. From a purely biochemical perspective, retaining the genetic information, the DNA and the enzymes, for synthesizing an unnecessary amino acid, especially if its synthesis pathway is complex, represents what biochemists sometimes call information of negative survival value.
Negative survival value. Because it costs energy to maintain.
Exactly. Even if the specific encoded genes are no longer actively expressed, the DNA itself has to be replicated, proofread, maintained, all of which requires precious cellular resources like ATP and nucleotides. Now, consider the energetic burden more directly. If you look at the number of enzymes required by, say, prokaryotic cells, which often retain these pathways and can be thought of as a proxy for our ancient ancestors.
Right, before they shed these abilities.
To biosynthesize the nutritionally essential amino acids versus the nutritionally non-essential ones, the difference is really quite striking.
Okay, I'm curious now. How many enzymes are we talking about here? Because if it takes a massive cellular factory just to produce one amino acid, that's a huge energy drain that could be better spent elsewhere, like you said.
It is indeed a significant disparity. Let's look at the numbers. To synthesize the entire suite of nutritionally essential amino acids, the ones we have to eat prokaryotic cells typically require a total of about 59 different enzymes. These pathways are often quite complex, involving many steps.
59 enzymes, okay.
Consider phenylamine, for example. Its biosynthesis pathway alone requires 10 distinct enzymatic steps. 10 enzymes just for that one.
Wow.
Now, compare that to the nutritionally non-essential amino acids, the ones we can make. To synthesize all of those, only about 17 enzymes are needed in total.
17.
Yeah.
Compared to 59.
Yep. And some are incredibly simple. Alanine, for instance, requires just one enzyme for its synthesis via transamination.
Wow, that's a huge disparity. 59 versus 17. That's a massive difference in metabolic overhead and cellular investment. It really highlights how much energy we save by simply eating these more complex compounds.
Absolutely. When we connect this to the bigger picture, it strongly suggests a strategic evolutionary trade-off for efficiency. It's as if our ancestors, over millennia, essentially decided, look, if this compound is readily available in our food and making it ourselves is biochemically complex and energy intensive, let's stop spending precious energy making it.
Makes sense.
Let's save that energy for other crucial processes, maybe reproduction, immune defense, brain development, or adapting to new environments. This allowed our lineage to shed complex, energetically expensive biosynthetic pathways in favor of acquiring these nutrients ready-made from the diet, assuming those nutrients were abundant in the available food sources.
It's a prime example of metabolic streamlining then, a highly optimized system for survival based on environmental availability.
Exactly, a very elegant evolutionary solution.
So what does this all mean for us today, or maybe more tragically, for communities around the world where diets are less varied and access to diverse food sources is a real challenge? It sounds like these distinctions, while fascinating biochemically, can have profoundly devastating real-world consequences, especially when that evolutionary bet on environmental abundance doesn't pay off.
They absolutely do. This is where abstract biochemical principles manifest as critical public health issues, demonstrating the direct link between molecular biology and, frankly, human suffering. Consider kwashiorkor.
I've heard of that, yes.
It's a severe nutritional disorder that tragically affects children, particularly in certain regions of, say, West Africa. It sometimes results when a child is weaned, perhaps too early, onto a starchy diet that is profoundly poor in high-quality protein.
So they're getting calories, but not the right building blocks.
Precisely. They're getting energy, maybe from cassava or maize, but critically lacking the necessary array of nutritionally essential amino acids, particularly ones like tryptophan and lysine, which are often low in those staples. The consequences are severe, often involving widespread edema, that visible swelling due to fluid retention skin lesions, changes in hair color and texture, and profound impaired growth and development. It's a stark picture of a body literally struggling to build and repair itself without its fundamental components.
That's heartbreaking. And I've heard of marasmus as well. Is that related? Or is it a different kind of deficiency that highlights the same underlying issues?
Marasmus is another form of severe malnutrition, but it typically occurs when there's a deficiency in both caloric intake and specific amino acids, as well as overall protein. So it's more like generalized starvation.
Total lack of resources.
Right. It's characterized by severe wasting of muscle and fat tissue. The child lacks both the energy and the amino acid building blocks for growth and repair, leading to that skeletal, emaciated appearance. Both kwashiorkor and marasmus tragically underscore how critical access to a complete protein profile containing all the essential amino acids in sufficient amounts really is. These disorders are sadly endemic in regions that rely heavily on staple grains, which, while providing vital energy, are often inherently poor sources of specific essential amino acids.
It's a powerful reminder of how fundamental diet is to health, right down to the molecular level, and how devastating the absence of just a few key components can be. And it's not just about broad populations, right?
Yeah.
Even individuals with specific medical conditions can face similar challenges due to absorption issues.
Precisely. That's a very important point. For instance, patients with short bowel syndrome, a condition where a significant portion of the small intestine is surgically removed, maybe due to disease like Crohn's or severely damaged, they struggle profoundly with nutrient absorption.
Because there's just less surface area?
Exactly. They're often unable to absorb sufficient quantities of calories and a broad spectrum of nutrients, including essential amino acids, from their diet, even if they eat well. This can lead to significant nutritional and metabolic abnormalities, mirroring, in some ways, the broader deficiencies seen in severe environmental malnutrition. But the cause is internal absorption failure rather than solely external food availability.
So they might need IV feeding.
Often, yes. It frequently necessitates intravenous feeding or parenteral nutrition to provide these vital compounds directly into the bloodstream, bypassing the damaged gut entirely.
Wow. This really raises an important question about global nutritional disparities and the fundamental life-sustaining role of accessing and absorbing a complete spectrum of these vital compounds. It highlights how quickly these complex biochemical principles manifest as critical health crises. It's not just a theoretical concept from a textbook.
Not at all.
It's literally a matter of life and death, reinforcing why understanding even these fundamental distinctions is so important for professionals in the field, from dietitians and doctors to global health policymakers. You need that deep understanding to tackle these issues effectively.
Absolutely. Foundational knowledge is key.
Now let's pivot a bit and get into the incredible biochemical symphony happening inside us. How our bodies actually make those nutritionally non-essential amino acids. It's really a masterclass in metabolic efficiency, isn't it? It illustrates the body's remarkable ability to synthesize what it needs from readily available ingredients.
It truly is an elegant system, and at the heart of much of this amino acid synthesis are enzymes known as transaminases, or you might see them called aminotransferases. Same thing.
Okay, transaminases, what do they do?
These enzymes play a fundamental and incredibly ubiquitous role in amino acid metabolism. Their main job is to catalyze the transfer of amino groups. Think of these as the defining nitrogen-containing part of an amino acid between an amino acid and an aceto acid.
So they swap the amino group.
Essentially, yes. Imagine them as molecular shuttles, precisely moving these nitrogen groups from one molecule, the amino acid, onto another molecule, the keto acid skeleton. This converts the keto acid into a new amino acid, and the original amino acid becomes a keto acid. And crucially, this is a reversible reaction.
Reversible, meaning it can go both ways.
Exactly, which means these enzymes can help build up amino acids when needed, or help break them down when they're in excess, depending on the cell's immediate needs. They're constantly working to maintain a delicate metabolic balance.
So they're kind of like the general contractors, moving vital nitrogen pieces around to build new structures or repurpose old ones. But it's not working alone, are they? You mentioned a symphony. What other key players are involved in this initial orchestration, especially around managing nitrogen?
That's a perfect analogy, general contractors. And no, they are indeed part of a larger coordinated team. The combined action of transaminases with two other crucial enzymes, glutamate dehydrogenase and glutamine synthetase, is what truly allows the body to efficiently incorporate potentially cytotoxic ammonium ions.
Cytotoxic, meaning harmful to cells.
Yes, very harmful, especially to the brain if levels get too high. Ammonium is a waste product of protein and amino acid breakdown. So these three enzyme types work in concert to manage this nitrogen waste efficiently. They essentially take a potential toxin, the free ammonium ion, and safely incorporate it into non-toxic amino acids like glutamate and glutamine.
So they're not just building, they're also detoxifying.
Precisely. It's a critical process for maintaining proper nitrogen balance throughout the body and preventing cellular toxicity. They are absolutely essential.
So these enzymes are like central metabolic hubs, orchestrating the initial steps for many other amino acids. And their ability to manage nitrogen is critical for preventing toxicity, as you said. It's impressive how the body takes a potentially harmful byproduct and converts it into something constructive and vital. It's a prime example of metabolic optimization.
It really is. Nature is incredibly efficient.
So if these are the assemblers, where do these non-essential amino acids even begin? What are their starting materials, their foundational building blocks within our cells? It sounds like we're not just plucking them out of thin air. There must be common precursors, right?
Absolutely not out of thin air. The body is incredibly resourceful, constantly recycling and repurposing molecules. The nutritionally non-essential amino acids are synthesized from common metabolic intermediates, primarily derived from two major energy-generating pathways that are constantly running in pretty much all your cells. The citric acid cycle, also known as the Krebs cycle.
Right, the central hub of energy metabolism.
Exactly. And glycolysis, the pathway that breaks down glucose. Think of these cycles as major biochemical highways, and these intermediates we use for amino acid synthesis are like crucial exit ramps leading to different biosynthetic destinations.
Okay, so which intermediates are we talking about?
Well, for instance, acetoglutarate. It's a central intermediate of the citric acid cycle, and it serves as the direct precursor for a whole family of amino acids. From acetoglutarate, our body can synthesize glutamate, glutamine, proline, and eventually hydroxyproline. It's a real cornerstone molecule for nitrogen metabolism.
Alpha-ketoglutarate, got it.
Yeah.
What about are there other major starting points?
Yes, definitely. Another key intermediate, also from the citric acid cycle, is oxaloacetate. This molecule is the precursor for aspartate and asparagine.
Okay, oxaloacetate for aspartate and asparagine. And from glycolysis.
From glycolysis, that central pathway for glucose breakdown, we have 3-phosphoglycerate. This molecule is the starting material for serine, and from serine, we can then make glycine. So these three intermediates, aketoglutarate, oxaloacetate, and 3-phosphoglycerate, are the prime powerhouses, the initial ingredients for building most of the nutritionally non-essential amino acids. It really showcases the deep interconnectedness of our metabolism, how energy pathways feed directly into building block synthesis.
Okay, let's delve into some of these specific biosynthesis pathways now, aiming for that mastery. Starting with glutamate. How does that one, which is so central a neurotransmitter, key in nitrogen metabolism, how does it come together?
Right. Glutamate, as we mentioned, is formed directly from aketoglutarate. This reaction is primarily catalyzed by mitochondrial glutamate dehydrogenase, an enzyme that lives inside our cellular powerhouses, the mitochondria.
And what's special about this enzyme?
Well, it's a remarkable enzyme because its activity strongly favors glutamate synthesis under normal physiological conditions, which is highly beneficial for the cell. And crucially, this reaction also plays that significant role we talked about in lowering the concentration of potentially toxic ammonium ions. It effectively pulls ammonia out of circulation and incorporates it safely into the usable amino acid, glutamate. So it's a vital detoxification step as well as a synthesis step. Dual function.
Very efficient.
Yep.
Okay, and then glutamine, which is so often talked about, for roles in gut health, immunity, even transporting nitrogen safely throughout the body.
Yeah.
How is that synthesized from glutamate?
Glutamine is synthesized from glutamate through an amidation reaction, meaning adding an amide group catalyzed by the enzyme glutamine synthetase. This process involves an interesting activated intermediate called glutamyl phosphate.
Activated, meaning it's made more reactive.
Exactly. Think of this enzyme, glutamine synthetase, as a sophisticated molecular assembler. It proceeds in a highly ordered fashion. First, glutamate and ATP, our energy currency, bind to the enzyme. Glutamate then gets activated by attacking a specific phosphorus atom on the ATP molecule, forming that glutamyl phosphate intermediate along with ADP. This activation makes the glutamate ready for the next step.
Which is adding the nitrogen.
Right. Next, an ammonium ion binds. Uncharged ammonia derived from the ion then attacks the activated glutamate intermediate. This leads to the formation of a temporary unstable structure. Finally, the release of inorganic phosphate allows for the release of the final product, glutamine. This intricate series of steps not only builds glutamine, but also demonstrates how the body invests significant energy, ATP, and enzymatic precision into creating what is essentially a safe nitrogen shuttle.
A nitrogen shuttle, I like that.
Yeah, it's critical for managing nitrogen balance and transporting it safely across tissues, for instance, from muscle, where protein breakdown might occur, to the liver or kidneys for processing or excretion. It's a testament to glutamine's critical role.
That's a lot of precise steps. It's fascinating how the body ensures such complex reactions proceed flawlessly. What about some of the maybe simpler ones, like alanine and aspartate? They seem to be constantly involved in metabolic reactions.
Their formation is comparatively straightforward, relying heavily on those ubiquitous transamination reactions we discussed earlier, catalyzed by transaminases. Alanine is formed directly from pyruvate, which is a key product of glucose metabolism, the endpoint of glycolysis, through transamination. Similarly, aspartate is formed from oxaloacetate, that citric acid cycle intermediate, also via transamination. In these reactions, an amino group is typically transferred from a common donor amino acid, often glutamate, onto the ketoacid skeleton, pyruvate or oxaloacetate, effectively converting that carbon skeleton into an amino acid.
So just a quick swap using a transaminase.
Pretty much. It's a quick, efficient, and reversible way to interconvert between these forms, responding rapidly to cellular needs.
Okay. And asparagine, how is that related to aspartate? The names sound very similar.
They are indeed closely related. Asparagine is synthesized directly from aspartate in a reaction catalyzed by the enzyme asparagine synthetase. This reaction actually bears a striking resemblance mechanistically to the glutamine synthetase reaction we just discussed. It also involves an activation step.
Activation with ATP again.
Yes, forming an intermediate called aspartyl phosphate. However, a key difference, at least in human systems, is that glutamine, rather than free ammonium ion, typically provides the nitrogen atom needed to convert aspartate to asparagine.
So glutamine acts as the nitrogen donor here.
Exactly. This highlights glutamine's broad role as a safe nitrogen donor throughout the body. The human pathway involves that aspartyl phosphate intermediate, and to ensure the reaction proceeds efficiently and is strongly favored towards synthesis, it's coupled with another reaction, the hydrolysis of pyrophosphate. pepane, into two molecules of inorganic phosphate by an enzyme called pyrophosphatase.
Why couple it like that?
Because breaking down pyrophosphate releases a good amount of energy. This energy release acts as a thermodynamic driving force, effectively pulling the asparagine synthesis reaction forward by removing one of its products, bipane indirectly, making the overall process essentially irreversible under cellular conditions. It ensures we make asparagine efficiently.
Clever. Using one reaction to power another. So similar mechanisms, but different nitrogen donors depending on the amino acid and even the organism sometimes, which has incredible adaptability.
Fascinating. What about serine, which is often linked to things like phospholipids and other crucial molecules maybe in cell membranes? Serine is synthesized from 3-phosphoglycerate, that key intermediate from the glycolysis pathway. So this links amino acid synthesis directly to how we primarily generate energy from glucose.
Okay, how does that conversion happen?
This synthesis occurs in three main steps. First, the O-hydroxyl group, an OH group, on 3-phosphoglycerate is oxidized, meaning it loses electrons to form 3-phosphohydroxypyruvate. This reaction is catalyzed by the enzyme 3-phosphoglycerate dehydrogenase and creates a reactive keto group, O.
Okay, step one is oxidation.
Step 2 is transamination. That keto group on 3-phosphohydroxypyruvate accepts an amino group, typically from glutamate, converting it to phosphoserine.
Step 2, add the amino group.
And finally, step 3 is dephosphorylation. An enzyme called phosphoserine phosphatase removes the phosphate group from phosphoserine, yielding the final product, L-serine.
Oxidation, transamination, dephosphorylation. Got it.
It's a neat three-step conversion from a glycolysis product, demonstrating again how the body cleverly redirects intermediates from one major pathway to build essential components needed elsewhere.
And glycine, the smallest amino acid, but surprisingly diverse in its roles, neurotransmitter, part of detoxification pathways. Where does that come from?
Glycine synthesis can actually occur through several important mammalian routes, which highlights the body's redundancy for making sure we have enough of these vital compounds. One way is via enzymes called glycine aminotransferases. These can catalyze its synthesis from a molecule called glyoxylate, using either glutamate or alanine as the amino donor. And interestingly, unlike many aminotransferase reactions, which are near equilibrium, this one is strongly favored for glycine synthesis, making it an efficient production pathway.
Okay, transamination from glyoxylate. Any other major routes?
Yes, significant amounts of glycine can also be formed from choline, which is an important component of lipids like phosphatidylcholine. There's a pathway involving several enzymatic steps that breaks down choline, ultimately yielding glycine.
From choline. Interesting.
But perhaps most importantly is its direct interconversion with serine. There's an enzyme called serine hydroxymethyltransferase. This enzyme can freely and reversibly interconvert serine and glycine.
So you can make glycine from serine and serine from glycine.
Exactly. It uses tetrahydrofolate, a crucial coenzyme derived from vitamin B9, folate, to handle the single carbon unit difference between them. This makes serine a direct and highly active precursor for glycine, ensuring a constant supply and balance between these two closely related amino acids.
Folate dependence there, that's important. Okay, what about proline? I know it's really important for the structure of collagen, that most abundant protein in our bodies. It has that unique ring structure.
It does, and that ring structure comes from its synthesis pathway, which actually starts with glutamate, again demonstrating glutamate's versatility as a precursor. The initial reaction involves converting the gamma-carboxyl group of glutamate to an activated intermediate glutamate phosphate, which sounds familiar, right?
Yes, similar to the glutamine synthesis step.
Very similar activation using ATP. This activated form is then reduced, it gains electrons to form glutamate semialdehyde. Now, this semialdehyde molecule has a reactive aldehyde group and an amino group positioned just right so that it undergoes a spontaneous cyclization. It snaps shut into a ring.
It just forms a ring by itself.
Spontaneously, yes, due to the chemistry. This ring structure is then further reduced in a final step to yield L-proline. So it's a pathway that beautifully illustrates how our bodies can transform a linear open-chain amino acid like glutamate into a cyclic one, proline, which gives it those unique structural properties so critical for proteins like collagen.
Activation, reduction, spontaneous cyclization, final reduction. Cool. Okay, now cysteine. I know you mentioned this one earlier. It's classified as nutritionally non-essential itself, but I remember you saying it depends on an essential amino acid. That still seems a bit counterintuitive. Can you unpack that dependency?
You're absolutely right to flag that, and it highlights a key nuance in the classification system that often causes confusion. While cysteine itself is nutritionally non-essential for humans because our bodies can make it, its synthesis is entirely dependent on having enough methionine, which is a nutritionally essential amino acid.
So no methionine in the diet means no cysteine synthesis.
Exactly. If you don't get enough methionine in your diet, your body won't be able to make enough cysteine, potentially leading to a cysteine deficiency even though it's non-essential. The pathway goes like this. Methionine is first converted through a series of steps involving S-adenosylmethionine, SANAM, into homocysteine.
Okay, methionine to homocysteine.
Then this homocysteine reacts with serine, one of the non-essential amino acids we just discussed, to form an intermediate called cystathionine. This step is catalyzed by cystathionine timothying.
Homocysteine plus serine makes cystathionine.
Right. And then, in the final step, another enzyme, cystathionine ileus, cleaves cystathionine to yield two products, cysteine and acetobutyrate, which gets metabolized elsewhere, releasing homoserine effectively.
So cysteine pops out at the end.
Correct. And what's fascinating here, if you track the atoms, is that the sulfur atom in the final cysteine molecule actually originates from the essential amino acid methionine, while the carbon skeleton of cysteine, its backbone, comes from the non-essential amino acid serine.
Wow, so it's a hybrid molecule in terms of its origins?
Precisely. It's a prime example of how metabolic pathways intersect and rely on both dietary essential and endogenously synthesized non-essential components in a highly intricate dance. It really underscores why just calling something non-essential doesn't tell the whole story.
That's a fantastic example. Okay, and tyrosine. Another interesting one because it's derived from phenylalanine, which we know is nutritionally essential. Does this mean if you lack phenylalanine in your diet, you can't make tyrosine either?
You've grasped a crucial point there. Yes, tyrosine is synthesized directly from phenylalanine by the enzyme phenylalanine hydroxylase. This means that if your diet contains adequate quantities of phenylalanine, then tyrosine effectively becomes nutritionally non-essential because your body can make all the tyrosine it needs from the dietary phenylalanine.
So adequate phenylalanine makes tyrosine non-essential.
Correct. However, and this is absolutely crucial, with major clinical implications, the phenylalanine hydroxylase reaction is irreversible.
Irreversible, meaning tyrosine cannot be converted back to phenylalanine.
Exactly. This means you cannot use dietary tyrosine to replace phenylalanine if you have a phenylalanine deficiency or an inability to convert it. This is starkly illustrated in the genetic condition phenylketonuria, or PKU.
Ah, PKU, I've heard of that.
In PKU, individuals have defects in the phenylalanine hydroxylase enzyme. They can't convert phenylalanine to tyrosine effectively. This leads to a dangerous buildup of phenylalanine and its byproducts, which are toxic, especially to the developing brain. But just as importantly, tyrosine becomes conditionally essential for these individuals. They must get tyrosine from their diet because they can't make it from phenylalanine.
So if you have PKU, eating tyrosine is critical, but it still won't fix the core problem of too much phenylalanine.
Precisely. The treatment involves a strictly controlled low phenylalanine diet supplemented with tyrosine. It really highlights the irreversibility of that enzymatic step.
What kind of enzyme is phenylalanine hydroxylase, and how does it work its magic of adding that hydroxyl group?
It's a type of enzyme called a mixed-function oxidase, or sometimes a monoxygenase. Think of it as a specialized chemical tool that uses molecular oxygen, O-uros, in a very particular way. It incorporates one atom from the O-butyl molecule into the para position, opposite side, of the phenylalanine ring, creating that hydroxyl group to form tyrosine.
And what happens to the other oxygen atom from oro?
Simultaneously, the enzyme reduces the other oxygen atom to form a molecule of water, HAHO. The reducing power for this reaction, the electrons needed for this chemical transformation, is provided by a crucial cofactor called tetrahydrobiopterin, BH4.
Tetrahydrobiopterin.
Yes, and BH4 itself ultimately gets its reducing equivalents, or electrons, regenerated using another cellular reducing agent called NADPH, which is vital for many biosynthetic reactions and antioxidant defense. So it's a whole electron relay system, ultimately linking back to our energy metabolism, all to perform this one specific hydroxylation. It's another testament to the elegant ways our body couples different reactions and pathways.
Incredible integration. And finally, just a quick note on valine, leucine, and isoleucine. These are the branched-chain amino acids, BCAAs, very popular in sports nutrition, important for muscle. They're nutritionally essential, we know that, but there's a metabolic nuance here too, isn't there, in terms of their interconversion with related molecules?
Indeed, even with the essential amino acids we must eat, there's still a layer of metabolic flexibility within the body. While leucine, valine, and isoleucine are all definitely nutritionally essential amino acids, our tissues, particularly muscle and liver, contain aminotransferases, those enzymes we discussed earlier.
The nitrogen swappers.
Exactly. These transaminases can reversibly interconvert these three branched-chain amino acids with their corresponding aikido acids. These are essentially the carbon skeletons of the BCAAs, but lacking the amino group.
So the body can swap the amino group on and off the BCAA skeletons.
Correct. This means that in a dietary context, if you were to consume their aikido acid forms, sometimes called keto analogs, your body could actually transaminate them, adding an amino group back on to synthesize the essential amino acids, leucine, isoleucine, and valine themselves.
So the keto acids could potentially substitute for the amino acids in the diet.
In theory, yes, and this has been explored therapeutically, for example, in kidney disease, patients needing to limit nitrogen intake. It demonstrates another layer of metabolic resourcefulness and interdependency, even for compounds we fundamentally need to acquire from our diet. The body can still do some final assembly if given the right precursors.
This whole section really illustrates the incredible interconnectedness of our metabolic pathways. Our bodies truly are master chemists, taking relatively simple ingredients derived from our core energy pathways and building these incredibly complex and vital amino acid molecules. And doing it all while managing waste like ammonia and optimizing for energy efficiency. It's a truly elegant system.
It really is. It allows us to synthesize what we need, often from readily available precursors, to support all the complex functions of life. It highlights the depth and sophistication of the body's internal factories, constantly working to maintain balance and build.
Okay, these next two amino acids are a bit of a curveball, a real departure from our usual thinking. They're incredibly prominent in our bodies, particularly in our connective tissues, but they have a fascinating and unique origin story. They aren't incorporated into proteins in the way we typically think, directly from a dietary source based on a specific genetic codon.
Ah, yes, you must be talking about hydroxyproline and hydroxylysine.
Exactly. Hydroxyproline and hydroxylase.
You're right, they're very special cases, and they showcase a different level of biochemical sophistication altogether. Hydroxyproline and hydroxylysine occur principally in collagen.
Which is everywhere in the body, right? Skin, bones.
Everywhere. It's the most abundant protein in mammals, providing structural integrity to everything from skin and bones to tendons, cartilage, blood vessels. It's essentially the molecular scaffolding that holds us together.
So what's unique about how they get into that collagen scaffold?
What makes their formation so unique is that there is no specific tRNA molecule, nor is there any specific codon in our genetic code that dictates their direct insertion into peptides during the initial process of protein synthesis, which we call translation.
No codon, no tRNA, so you can't just read them off the mRNA.
Nope. This means you could eat pure hydroxyproline or hydroxylysine, but your body wouldn't just slot them into a growing protein chain on the ribosome during translation. It doesn't work that way for these two.
So if they're not put in during the initial protein building based on our genetic code, how do they get there? Is it like an after-the-fact modification, a kind of specialized finishing touch?
Precisely. That's exactly what it is. This is a classic example of what we call a post-translational modification, meaning it happens after translation, after the initial protein chain is made.
Okay, post-translational. Makes sense.
Peptidyl hydroxyproline and peptidyl hydroxylysine and peptidyl just means they're already part of a peptide or protein chain. These modified amino acids arise from regular proline and lysine residues only after these original amino acids have already been incorporated into the nascent collagen peptides by the ribosome following the standard genetic code.
So the collagen chain gets built with normal proline and lysine.
Correct. And then specific enzymes come along afterwards and modify some of those prolines and lysines, adding a hydroxyl OH group. It's like building the basic house structure first, using standard bricks, and then adding very specific, highly specialized reinforcements later, the hydroxylation, to ensure the final structure is strong and stable. This modification is absolutely critical for collagen's strength and stability.
And what are the enzymes and, importantly, the cofactors involved in this crucial finishing work? Because I know this is where some really well-known dietary deficiencies can come into play, often with very visible consequences.
Yes, this is where the biochemistry ties directly and quite dramatically to nutrition and human health. The hydroxylation of these peptidyl, prolyl, and peptidyl lysyl residues is catalyzed by specific enzymes.
Prolyl hydroxylase and lysyl hydroxylase, respectively.
Prolyl hydroxylase and lysyl hydroxylase. These enzymes are found abundantly in tissues where collagen synthesis is high, places like skin, skeletal muscle, and especially in granulating wounds during healing, because strong collagen is absolutely essential for tissue integrity and repair.
And what do these enzymes need to do their job? The cofactors.
Right, this is critical. These hydroxylases require a very specific set of crucial cofactors to function properly. They need molecular oxygen, ascorbate, which is vitamin C.
Ah, vitamin C, there it is.
Yes, the famous one. They also need ferrous iron, Fa, and IK-glutarate, that citric acid cycle intermediate we met earlier.
Oxygen, vitamin C, iron, and alpha-ketoglutarate. Quite a list.
It is. The enzymes themselves are classified, like phenylalanine hydroxylase, as mixed-function oxidases, or dioxygenases. What this means mechanistically is that for every molecule of proline or lysine that gets hydroxylated, gets an OH group from O, one molecule of iketoglutarate is simultaneously decarboxylated, loses COs, to become succinate. The other oxygen atom from OOs goes into the succinate molecule.
So it's a coupled reaction involving oxygen and alpha-ketoglutarate.
Exactly. It's a beautifully coordinated chemical dance that ensures the hydroxylation reaction is energetically favorable and proceeds efficiently, linking amino acid modification back to central metabolism again.
And this brings us directly to one of the most classic nutritional deficiency diseases, scurvy. Suddenly, it's not just about not getting enough vitamin C. It's about a specific, critical biochemical reaction grinding to a halt. Puts a whole new perspective on those historical tales of sailors suffering terribly on long voyages.
Exactly. The clinical presentation of scurvy is quite distinct and frankly grim. You see bleeding gums, loosening teeth, swelling and pain in the joints, poor wound healing, easy bruising, corkscrew hairs, and eventually anemia and profound fatigue. The direct biochemical explanation for these symptoms is a severe deprivation of vitamin C, ascorbate.
Because the hydroxylases can't function without it.
Precisely. Without sufficient vitamin C acting as a crucial cofactor, likely keeping the iron atom in the correct reduced state, these two vital enzymes, prolyl hydroxylase and lysyl hydroxylase, cannot function effectively. This results in impaired hydroxylation of the peptidylproline and peptidylysine residues within the newly synthesized collagen chains.
So the collagen is made, but it's faulty.
It's unstable. Without these vital hydroxylations, the collagen triple helix cannot form properly at body temperature. It becomes structurally unsound, like trying to build a house with weak, wobbly beams. This also leads to a failure to provide the necessary hydroxylated lysine residues that are substrates for the subsequent cross-linking reactions needed to strengthen the mature collagen fibers. In essence, the structural integrity of connective tissues throughout the entire body is profoundly compromised.
Which explains all those symptoms, the weak blood vessels causing bleeding, the poor wound healing.
Exactly. It all comes back to unstable collagen. In severe, untreated cases, scurvy can unfortunately lead to death, illustrating the absolute necessity of a single vitamin for a specific, critical post-translational modification.
It's incredible how a single nutrient deficiency can have such widespread and devastating effects simply by disrupting these specific molecular modifications, making our very physical framework unstable. Are there also genetic disorders related to this crucial process? Things that aren't dietary?
Yes, absolutely. Beyond dietary deficiencies like scurvy, genetic disorders of collagen biosynthesis also exist, highlighting just how critical the proper formation and modification of this protein is. For example, several forms of osteogenesis imperfecta, sometimes known as brittle bone disease.
Right, where bones fracture very easily.
Exactly. These often result from mutations in the genes encoding the collagen chains themselves, or sometimes in the genes for enzymes involved in collagen processing and modification. Similarly, Ehlers-Danlos syndrome is actually a group of diverse inherited connective tissue disorders.
What happens there?
It typically results in symptoms like overly mobile, hypermobile joints, highly elastic or stretchy skin, and fragile tissues, including blood vessels. These syndromes can arise from defects in various genes involved in collagen formation or processing, including, in some types, defects in lysyl hydroxylase, one of the very enzymes we've been discussing.
So genetic defects can mimic some aspects of the nutritional deficiency.
In a way, yes. They both lead to faulty collagen. These genetic conditions underscore that the precise assembly, modification, and cross-linking of collagen are absolutely non-negotiable for human health. Any weak link in that chain, whether dietary or genetic, can have severe consequences.
This really highlights how intricately our nutrition, our genetics, and our biochemistry are interwoven. A single dietary deficiency, like lacking vitamin C, or even a tiny mutation in a single gene, can profoundly compromise the very framework that holds our bodies together. It underscores why understanding not just what happens, but why these modifications occur and what they require is so crucial for health professionals. It helps them get beyond just treating symptoms to potentially addressing the underlying biochemical dysfunction. It's about achieving that elite level of professional insight, right?
Precisely. Understanding the mechanism gives you the power to intervene more effectively.
And finally, let's talk about a truly surprising one, sometimes called the 21st amino acid, which is itself a testament to the incredible flexibility and, well, ingenuity of our genetic code. We've talked about the standard 20, but what makes selenocysteine so special? And how does our body manage to incorporate something so rare and vital into its proteins, especially since it contains selenium, a trace element that can be toxic if you get too much?
Selenocysteine is indeed a marvel, and its existence and incorporation truly demonstrate the unparalleled sophistication of our cellular machinery. While its occurrence in proteins is relatively uncommon compared to the other 20, we currently know of at least 25 human selenoproteins, maybe more.
Only 25 or so.
Around that number identified so far, yes, but its scarcity really belies its immense importance. Selenocysteine is crucially present right at the active site, the business end of several vital mammalian enzymes. These enzymes typically catalyze redox reactions.
Reactions, meaning electron transfers.
Exactly. Reactions involving the precise transfer of electrons, which are essential for countless cellular processes, including energy production, signaling, and especially antioxidant defense.
So it's not just present in these enzymes, it's actively involved in their work, almost like a specialized chemical tool.
Precisely. It directly participates in the catalytic mechanism of these enzymes. Selenium has unique chemical properties different from sulfur and cysteine that make it highly reactive and particularly suited for certain types of redox catalysis. Examples of important human selenoproteins include thioredoxin reductase.
What does that do?
It plays a key role in antioxidant defense and also in providing building blocks for DNA synthesis. Then there's glutathione peroxidase, another family of key antioxidant enzymes that protect our cells from damage caused by reactive oxygen species like hydrogen peroxide.
Okay, antioxidant defense seems key. Any others?
Yes, another critical one is the dehydrogenase enzyme family. These enzymes convert the thyroid hormone thyroxine, T4, into the much more active form, triiodothyronine, T3. This conversion step, which involves removing an iodine atom, is absolutely critical for regulating metabolism throughout the body, and it relies on selenocysteine at the active site.
Wow, so thyroid function depends on it too. Is it really better than cysteine for these jobs?
Often, yes. The fact that replacing the selenocysteine residue with a regular cysteine residue, which is chemically similar, just sulfur instead of selenium in these enzymes, can actually reduce their catalytic activity significantly, sometimes by orders of magnitude, really underscores selenium's unique and often irreplaceable properties in these specific high-performance roles. It's literally tuned for optimal function in these contexts.
And if something so vital is impaired, I imagine there are serious clinical implications that reach far beyond just, say, a simple trace element deficiency showing up on a blood test.
Absolutely. Impairments in human selenoprotein function, whether due to genetic defects or severe dietary selenium deficiency, have been directly implicated in some very serious health issues. These include links to tumorigenesis, the complex process of cancer development and atherosclerosis, the underlying cause of heart attacks and strokes.
And isn't there a specific heart condition linked to selenium deficiency?
Yes, a notable and quite tragic example is selenium deficiency cardiomyopathy, known as Keyshawn disease. It primarily affects children and women of childbearing age in regions with extremely low dietary selenium intake, historically certain parts of rural China where the soil's selenium content is exceptionally low. It directly affects the heart muscle, leading to an enlarged heart and potentially severe heart failure. It's a stark reminder of the essential nature of this trace element and its unique amino acid derivative, illustrating how a micronutrient deficiency can lead directly to macro-level organ failure.
That's a powerful connection between a trace element, a unique amino acid, and major human diseases. But how does the body even synthesize selenocysteine? And maybe more amazingly, how does it incorporate it into proteins when it's not one of the standard 20 and its designated codon, UGA, normally means stop he? This is where it gets really mind-bending, isn't it? Almost like a cellular magic trick.
This is indeed where the truly extraordinary biochemistry comes into play, revealing an ingenious workaround in the standard interpretation of the genetic code. The biosynthesis of selenocysteine itself requires several components. You need serine, which provides the carbon skeleton. You need selenate, SCO, the inorganic form of selenium usually obtained from our diet. You need ATP for energy. You need a very specific, unique type of tRNA molecule, different from all others. And you need several specialized enzymes.
Okay, so this starts with serine and selenium gets added somehow.
Sort of. The actual selenium donor molecule used in the process is something called selenophosphate. This is synthesized first from ATP and selenite via an enzyme called selenophosphate synthetase. This reaction essentially activates the selenium, making it ready for incorporation.
Activated selenium, got it. Now the mind-bending part. How does it get into the protein?
Right. The truly fascinating part, the co-translational insertion marvel, is how it gets into proteins. Unlike hydroxyproline or hydroxylysine, which are added after the protein is fully built, that post-translational modification.
Right.
Selenocysteine arises co-translationally. This means it's inserted during the protein synthesis process as the ribosome is actively reading the messenger RNA, mRNA, and building the polypeptide chain. This makes it function essentially like a 21st amino acid during translation itself.
Co-translational during translation. So it's actively being built into the chain as it's synthesized, not just tacked on later. But if it's codon, UGA normally signals stop P to the ribosome, telling it to terminate protein synthesis. How on earth does the machinery know to put in selenocysteine instead of just stopping? That seems like a massive potential error point in such a precise system.
That's the absolute crux of the matter, and the cell employs an elegant solution that showcases the extreme precision of cellular regulation. The protein synthetic apparatus identifies a selenocysteine-specific UGA codon, distinguishing it from a UGA codon that genuinely means stop through a unique mechanism. This involves a special structure in the mRNA molecule itself, called the selenocysteine insertion element, often abbreviated as CSIS.
Dances element. What's that?
It's a specific stem loop structure, basically a distinctive folded shape, usually found in the 3' untranslated region, 3-UTR, of the selenoprotein mRNA molecule, typically downstream from the UGA codon itself. This SESIS element acts like a special signal flag or perhaps a specific docking site on the mRNA. It physically signals to the ribosomal machinery and associated factors, hey, don't stop here. At this specific UGA codon, in this particular mRNA context, you need to insert selenocysteine.
So the context provided by the CISI comment overrides the default meaning of UGA.
Exactly. It allows for context-dependent recoding of the UGA codon. Now, how does the selenocysteine actually get delivered? That involves the specialized tRNA we mentioned called tRNA-seq.
The selenocysteine tRNA.
Right. Now, tRNA-seq is unusual. It's first charged or loaded with serine by the same enzyme, ligase, that normally charges the standard serine tRNAs, tRNaser. So initially, tRNA-seq actually carries a serine molecule.
Starts with serine, not selenocysteine.
Correct. Then, a series of subsequent enzyme-catalyzed reactions take place right there on the tRNA molecule. These reactions convert the oxygen atom of the attached serine into a selenium atom, using that activated selenophosphate we mentioned earlier as the selenium donor. This chemical transformation converts the serenal tRNA-seq into aminoacryl tRNA-seq and finally into selenocysteine tRNA-seq. Now it's carrying selenocysteine.
Wow, the modification happens on the tRNA itself.
Exactly. Finally, a specific specialized elongation factor protein, distinct from the usual elongation factors like EF2 in bacteria or EF1A in eukaryotes, recognizes this unique selenocysteine tRNA-seq. Guided by the interaction with the SESIS element on the mRNA, this special factor delivers the selenocysteine tRNA-seq to the ribosome, allowing the selenocysteine to be precisely incorporated into the growing protein chain at the UGA codon. The normal stop factors are prevented from binding. It's a truly masterclass in contextual genetic code interpretation and specialized molecular machinery.
If we connect this to the bigger picture, selenocysteine is just an astonishing testament to the intricate and highly regulated mechanisms our cells use to achieve specific high-performance functions. It's an extraordinary example, like you said, of how the genetic code can be reprogrammed or reinterpreted in a specific context, allowing a typically stop signal to instead insert a vital, specialized amino acid. That level of precision, adaptability, and context-dependent instruction in our molecular machinery is truly astounding.
It really is.
It raises an important question for professionals aiming for that deep mastery. What other subtle regulatory mechanisms might be at play in our bodies, perhaps even more complex, that we are only just beginning to uncover? What other ingenious designs are waiting for us to figure them out?
Exactly. It definitely makes you wonder how much more there is to learn about these finely tuned processes that allow for life to function at such a complex, yet seemingly efficient level. It constantly pushes the boundaries of our understanding of molecular biology and genetics.
Well, this deep dive has truly been a comprehensive journey, hasn't it? Really getting into the weeds of understanding amino acids. We've gone from that critical distinction between what our bodies must eat, the nutritionally essential, and what they can make themselves, the nutritionally non-essential.
Making sure we understand all 20 are metabolically vital.
Absolutely. Then we move through the complex yet elegant pathways of how our cells actually synthesize those non-essential compounds. And then we ventured into the fascinating specialized roles of amino acids like hydroxyproline and hydroxylysine, modified after synthesis to give collagen its incredible strength.
The post-translational modifications.
Right. And finally, we explored the incredible story of selenocysteine, the 21st amino acid, inserted during the protein building process, elegantly defying a typical genetic code signal through that amazing co-translational mechanism. I feel like we've genuinely achieved that mission of really digging deep to master this information, thinking about it like the elite professionals in this field would.
I agree. We've seen how every single one of the 20 common amino acids is metabolically essential for human health, even if only a subset must be obtained directly from the diet. The body's remarkable ability to synthesize the nutritionally non-essential ones from readily available metabolic intermediates through these sophisticated enzymatic pathways is a profound testament to its metabolic efficiency and resilience.
Definitely.
And the discovery of these highly specialized roles for modified amino acids, like their critical functions in collagen stability with hydroxyproline, or as essential catalytic components of vital redox enzymes with selenocysteine, highlights the profound and intricate layers of biochemical regulation that underpin our very well-being. This level of understanding provides a really critical framework for comprehending both health and disease at a molecular level.
This deep dive has really shown us that every bite we take and every single molecular reaction happening within us right now is part of an incredibly sophisticated orchestra, playing out this complex symphony of life. It makes you wonder, doesn't it, what other hidden players are out there, quietly orchestrating our health in ways we're only just beginning to grasp.
That's a great thought.
What other subtle chemical signals or unique molecular structures are yet to be fully understood, just waiting to reveal their secrets and potentially transform our understanding of medicine or nutrition?
Absolutely. And building on that, this raises an important question for the future. How does our increasing understanding of these minute biochemical details, this mastery we've discussed, truly empower us to make better decisions about nutrition and health, not just individually, but on a grand scale?
Right. What does it mean practically?
What does this level of mastery imply for the future of personalized medicine, for instance, where we might one day tailor diets or therapies based on an individual's unique genetic predispositions for amino acid synthesis or transport or modification? And what does it mean for our collective well-being as we continue to connect these incredibly complex dots between our diet, our genes, and our molecular function?
The potential seems enormous.
It really does. The more we understand these fundamentals at this deep level, the more precisely and effectively we can potentially intervene to prevent or treat disease, truly pushing the boundaries of what's possible in healthcare.
It's an exciting thought, isn't it? The more we uncover about these fundamental building blocks of life, the more we appreciate the extraordinary complexity and elegance that makes us who we are. So we encourage you, our listeners, to keep that curiosity alive. What other seemingly simple biological processes do you think hold such profound complexity? Keep exploring, keep learning, and keep diving deep into the world around and within you.